Right around the 4th of July, the sockeye salmon run peaks in Bristol Bay, Alaska. The commercial catch pours into remote facilities, like the Naknek cannery, pushing tens of millions of fish down industrial packing lines in a matter of weeks. The sheer volume of biological mass moving through this system is staggering. During a summer shift, a teenager working the canning line might step outside and see grizzly bears standing right at the chain-link fence, drawn by the immense physical presence of the run. Looking at this image of a dense sockeye cluster, you can see the problem immediately. The entire multi-billion-dollar industrial throughput relies on knowing the biological status of these fish, but separating and counting individual bodies in this chaos is nearly impossible. This map illustrates the core observational constraint. Salmon are anadromous. They hatch in fresh water, but spend up to 99% of their lives circulating through the massive, untrackable expanse of the North Pacific Gyre. That ocean is a biological black box. Because we lack the instruments to track a scattered population through thousands of miles of open saltwater, fisheries managers have to wait. They wait for the adult fish to navigate back to the coast and push up their natal freshwater streams to spawn. We cannot measure the run in the ocean. We have to capture the data at the absolute narrowest point of their migration, turning a vast marine mystery into a highly localized sensor problem. This weir demonstrates exactly what that choke point looks like. As fish move upriver, managers must calculate escapement, the exact number of spawners allowed to safely bypass the commercial fishing nets so they can reproduce. The target is a highly specific window called the biological escapement goal. Let too few fish escape, and future runs collapse. Let too many slip through, and the excess spawners crowd the gravel riverbeds, dying without successfully reproducing. Here you see the economic consequence, a massive commercial fleet waiting in the water. The operational window for these boats is dictated hour by hour, based entirely on the upriver escapement data. The instrument used to calculate that escapement is rapidly moving from a human with a hand clicker to an artificial intelligence model. That shift redefines our understanding of salmon biology, and it pushes the limits of what off-grid computing can actually achieve. Counting escapement is the central feedback loop of Bristol Bay. It is the physical throttle determining the survival of the ecosystem and the economy built on top of it. The baseline for this counting system was established in 1955. Alaska Department of Fish and Game technicians climb aluminum scaffold towers erected on the riverbank, put on polarized sunglasses to cut the glare, and manually tally the fish using mechanical hand clickers. This aerial photo captures the geographical extreme of the job. These human sensors are deployed to isolated, off-grid field camps surrounded by roadless wilderness, subjected to mosquitoes, fatigue, and 24-hour rotating shifts. This equation breaks down the human sampling method. Staring at moving water for 8 hours straight destroys concentration. Instead, technicians count passing fish for exactly 10 minutes of every hour, and then multiply that number by 6 to estimate the total hourly passage. Despite relying on human eyes and a pocket clicker, this statistical extrapolation works. Well-run tower projects consistently yield error rates between 6 and 10%. The constraint lies in the biology of the observer. The system immediately fails the moment the river rises, the water turns turbid with mud, or the ambient light drops too low to penetrate the surface. The manual tower operates as a highly effective 70-year-old statistical instrument. Any automated camera or algorithm looking to replace it has to prove it can beat that established baseline. The first attempt to beat that baseline relies on optical cameras. Researchers deploy self-contained drones in a box and submerged lenses to provide continuous, non-intrusive video monitoring of the riverbed. To train the software, a human technician sits at a laptop and watches hours of this drone footage. Every time a salmon crosses a virtual yellow line on the screen, the technician presses a key, generating thousands of labeled examples to teach the machine what a fish looks like. Drawing a bounding box in an isolated frame is just object detection. Without tracking stitching frames together, a camera counts the exact same fish 60 times. The architecture assigns a persistent ID tag, ensuring one organism equals one tally. In a controlled lab, the logic is flawless. On a riverbank, the algorithmic logic shatters against the chaotic physics of moving water. Surface glare strokes the lens with bright white blobs. clouds shift across the sun, darkening the entire riverbed. Heavy rain pushes silt downstream, rendering the targets completely invisible. Then there is the drifting stick hallucination. Tumbling waterlogged debris mimics the exact size, shape, and motion profile the model associates with a salmon. A human ignores it instantly. The machine logs a false positive. Optical computer vision relies heavily on pattern recognition. Wild environments constantly generate adversarial patterns that easily fool an algorithm, but which biological human eyes filter out by instinct. To solve the optical failures, agencies turn to imaging sonar. Devices like the ReThink acoustic camera blast high-frequency sound waves across the riverbed, using echoes to build high-resolution outlines of fish, regardless of glacial silt or pitch-black darkness. Sonar guarantees you can see the target, but running multiple acoustic cameras around the clock generates hundreds of hours of massive, three-dimensional video files. The state-of-the-art method for analyzing that data uses heavyweight models like YOLO V5M paired with a tracker. It relies on over 21 million parameters to assess a single frame. This matrix shows the resulting edge compute crisis. Processing 21 million parameters 60 times a second demands high-end graphics processing units and a grid-level electrical supply. A field technician on the riverbank has a consumer laptop and a portable solar array. The researchers are operating out of canvas tents. They have limited battery reserves and absolutely zero high-speed internet to offload those massive video files to a cloud data center. By utilizing sonar to solve the biological visibility problem, the engineers instantly created an edge computing crisis. Crucial management data is now trapped inside machines that lack the local power to process it. The engineering challenge requires finding a way to process millions of frames of acoustic data locally, on a cheap laptop, without a dedicated GPU. The breakthrough is temporal data compression. Instead of analyzing the sonar as a continuous, heavy movie file, the software flattens time into space. Watch this frame of sonar data. The system locates only the brightest echo intensity, the strongest return from a solid object, and extracts it into a single 1-pixel-wide vertical strip. It then sequences 200 of these vertical strips side by side. Time becomes the horizontal x-axis. A heavy video clip is squashed down into a single static 2D image map called an echogram. In this compressed two-dimensional space, a fish swimming through the sonar beam no longer looks like a fish. It registers as a continuous diagonal streak, turning the entire run into a scannable barcode or a long-exposure photograph of highway traffic. The operational payoff is massive. Instead of running a heavy 21 million parameter model 200 separate times, a lightweight image recognition model, ResNet 18 at just 11.7 million parameters, glances at the echogram image exactly once. The ResNet model stops trying to track individual fish entirely. It simply reads the slope and density of the streaks, directly outputting two numbers, the upstream tally and the downstream tally. This compression method requires a direct trade-off. The heavy model boasts an error rate near 5%. The echogram currently sits between 23 and 30% error, but it operates effortlessly on basic edge hardware. In remote deployments, a perfect model that requires a data center provides 0 practical counts. An imperfect model running natively on the riverbank delivers the data managers need to make decisions today. Scaling these automated sensors is biologically critical because the baseline stability of the North Pacific is degrading. While Bristol Bay sockeye have recently experienced an explosive population boom, Chinook and chum runs in the Yukon, just hundreds of miles away, have suffered catastrophic collapses under the same climate pressures. This chart compares historical and modern body sizes. A 2020 study confirms that salmon are returning from the ocean younger and smaller. Mean body mass dropped by 8% for Chinook and 2.1% for sockeye. A smaller fish carries fewer eggs. Counting individual bodies the traditional way increasingly overestimates the actual reproductive capacity arriving at the spawning grounds. The runs are also facing lethal temperature spikes. In 2019, a severe riverine heat wave pushed water temperatures past biological thresholds, triggering widespread pre-spawn die-offs across thousands of miles of Alaskan habitat. As the climate signal grows more chaotic, managers can no longer bet the season on extrapolated 10-minute human samples. They require continuous, high-fidelity monitoring to spot rapid ecosystem degradation before it locks in. This vast Alaskan watershed supports 15,000 jobs and generates $2.2 billion annually. It is also the site of ongoing federal litigation over the proposed Pebble Mine, a massive copper and gold deposit situated directly at the headwaters. As AI models and drones slowly take over the official escapement counts, a macro-political question surfaces. Who owns the model and who controls the resulting data? Holding the proprietary algorithm that counts the salmon effectively grants the holder the power to define the precise biological and economic value of the river in a court of law. Back at the Naknek cannery line, the July peak arrives regardless of the politics. The grizzlies stand at the fence, and the teenagers pack the catch. Whether the final escapement count is manually clicked by a technician on a cold aluminum scaffold, or processed in a fraction of a second by a ResNet 18 model reading acoustic barcodes, the biological mandate does not change. The fish must return home. The tools of observation will continue to evolve, moving further toward the edge of compute capability, but that single escapement number remains the absolute fulcrum upon which the survival of the species, and the humans who rely on it, balances.